final plenary speaker, Joachim. He's recently retired from the US EPA after 32 years as a research scientist focusing on human systems biology of exposure. He's an adjunct professor at the University of North Carolina. He's published over 150 journal articles, many involving breath biomarker research and statistical interpretation of breath-based data. He's the co-editor of the recent Elsevier book, Breath-Borne Biomarkers, the Human Volodilome. He currently serves as editor-in-chief at the Institute of Physics Journal of Breath Research, consults with the US NASA projects on jet fighter pilot breathing assessments, and International Space Station air quality. He's a founding member of the International Association of Breath Research. Over to you, and thanks for joining us today.
Good afternoon. Today, I want to talk about breath research, the Journal of Breath Research, and how COVID-19 is affecting our breath research community. We, the co-authors of this talk, represent the editors of the Journal of Breath Research. As we can't be there today, I have taken the liberty of putting some pictures of us up. The one on the upper left is me, Joachim Pleil. Next to me is Jonathan Beecham from Fraunhofer Institute in Freising, Germany. And in the second row are Raed Dwight from the Cleveland Clinic and Terence Risby from Johns Hopkins University. So as background for this talk, we as the editorial staff realize we have the opportunity and also the responsibility to contribute to the global public health, especially with respect to COVID-19, which number one is a respiratory illness for the most part, and number two is spread via exhaled breath/aerosols. So these topics fit in well with the journal. And we want to foster and publish research that will ultimately help diagnose, treat, and cure the COVID-19 disease. In doing this, we also realize that we're in a new world of breath research with respect to safety. Breath is no longer the innocuous biological fluid that is just spent into the air. We now have to be careful about infection and treat breath more like blood and urine than we have before. So what we're doing at the journal is, number one, trying to encourage topics related to exhaled breath condensate, and aerosols, expanding the scope of JBR to include pulmonary function as a dependent variable. This refers to the physics of breath, flow, pressure, timing, volume, in addition to our traditional staples of, chemistry of breath or analytical techniques or sampling methodology. We are trying very hard to fast track COVID-19 papers and doing more outreach to get groundbreaking papers to be submitted to the journal. We'd like to think of the journal as kind of the centroid for breath research regarding COVID-19. The other thing we're doing is starting to require disclosure of safety protocols. New submissions are now asked to provide some text regarding how they deal with safety in the laboratory, how they deal with breath cleanup, breath instrumentation, avoidance of cross-contamination, and also protection of subjects, patients, researchers, and medical staff. We are asking new submissions to provide this information analogous to, say, an IRB kind of statement regarding human studies. We all understand breath research has changed in the COVID era. We have to protect the staff, the subjects, the patients, the researchers from contamination and infection. We are now looking more closely at medical ventilators, improving, modifying, etc., and using medical ventilators. We found that during the initial onslaught in New York, for example, the medical ventilator was the one main tool for preserving life with people with critical illness from COVID. We are also fostering a new emphasis on exhaled aerosols, just in general, and also specifically because the aerosol seems to be the primary vector of cross-contamination and infection. We are taking a closer look at developing preclinical diagnostics using breath, potentially as a replacement or complement to the nasal swabs and other swabbing methods. We're looking into the issues regarding the restriction of viral spread. Social distancing really isn't on our radar, so to speak, but the use of masks and improvised facial coverings definitely is. And finally, we're also taking a very close look at analytical technology, especially with respect to identifying new chemicals in breath. We would ultimately like to get some biomarkers in breath that could help us diagnose disease, but it would also be very helpful to know, if we find some candidates, what they are. We have instituted a new requirement for peer review of submissions at JBR. And the idea is, as I said before, to require people to really think about how they're dealing with potentially infectious issues in their laboratories or clinics. The new instructions are available in the hot link from the website, but for you, I have extracted some of the text about this. And so basically what we're saying is, when you submit a new article, we would like for you to discuss in a short statement your personal protective measures, the safety of your sampling equipment, safety procedures for the analysis of collected samples, etc. We're trying to be very proactive at the journal, and hopefully this will have some value when people are writing, if they keep in mind that they're going to have to explain how they're doing things, and maybe it will help upgrade some of these safety measures in the various laboratories. So the medical ventilator came onto the forefront, not because of how it works, but its lack of access. If you recall, it was major media news pretty much everywhere there was a COVID outbreak, that basically at the time, the only good method we had of preserving life of critically ill patients was to put them on a medical ventilator. At that time, we did not know enough about COVID-19 to have some of the regimen of antiviral drugs and treatments, whatever that we have now. And the first step was put someone on a ventilator and hope for the best. So there was a shortage of ventilators. And so the whole gestalt of ventilators became big time news. We tried to get more of them made quickly. And so the understanding and modification of medical ventilators is still kind of a big area of thought during the COVID crisis. We have to realize medical ventilators are complicated. They have to provide flow, pressure, volume, and timing, the physics of breath, in a way that preserves life to patients who cannot breathe for themselves. And so they have to be very well adapted to any individual patient. Using these is non-trivial, and using them correctly, that is. But from a research perspective, the medical ventilator actually presents itself as basically a sampling tool. If you think about it, as you are ventilating the patient, breath flows in and the breath flows out. And as it flows out, it gives opportunity to collect sample. Imagine that you tried to collect sample from critical ill patients via blood. It's much more difficult, but they're breathing anyway. And so just tapping into the exhale stream of a ventilator is a good opportunity to collect samples to track health state, just see about recovery and such. We, in the global we, some of our colleagues have been working on collecting EBC, which is kind of a waste product of a medical ventilator. Imagine the exhaled leg of the ventilator stream is humid and hot, and it drops out EBC on a continuous basis. My good friend and colleague Mike Davis, Indiana University, has been working with medical ventilators for a long time and has now developed ways of extracting breath condensate from medical ventilators without interrupting the ventilation loop. So this has turned out into quite a boon for tracking and understanding what is going on in the breath of critically ill patients. Mike and company have recently written a journal article, a Journal of Breath Research, about breath condensate in critically ill mechanically ventilated patients. This is going to be published very soon. It's already in the accepted manuscript stage. I've put reference to it at the bottom of this page. Another area that we're looking into very strongly now is EBA, or exhaled breath aerosol. The aerosols are a small fraction of the total breath. As you know, breath is mostly gas. Its job is to exchange carbon dioxide and oxygen in the blood, gas. However, because of the nature of the lungs, we do exhale a fair amount of aerosol. It's a small relative amount. We exhale about somewhere around 1 milliliter of exhaled breath condensate, which contains the aerosol, in 10 to 15 minutes of breathing. So it is a fair amount of water that comes out. The interesting thing about exhaled breath aerosol is that it provides basically a non-invasive window for diagnosing infection. Because the infectious materials are thought to be encapsulated in these little wet droplets that we call aerosol, we have now come to the conclusion that we can trap these on masks and other surfaces, respirator surfaces, non-rebreathing gear, things like that. And as these wet little aerosols are traveling from the mouth out and crash into surfaces, they tend to stick, whereas the gas, of course, continues to move because the molecules are much smaller and lighter. This issue of collecting aerosol serves two purposes. One, it makes whatever surfaces we're crashing them into, be it masks, scarves, ventilators, respirators, whatever, they make good sampling media. We have done some work with firefighters, and we've done some work with jet fighter pilots who wear these on-demand breathing masks, and we have literally swabbed exhaled aerosol out of the insides of the mask and used them as an analytical tool to see what they've been exposed to, or to see what possibly their inflammatory status is. So that's part one. Part two is, by virtue of the fact that the aerosols are crashing into these surfaces and staying behind, those aerosols are no longer flying around in the air beyond the person. And so trapping EBA with masks, which we know works because we can analyze trapped EBA, serves as a protective measure against spreading the virus as well. So to kind of lay this out in some couple of text styles, we've written two editorials recently, one predating the COVID crisis, where we talk strictly about how the exhaled breath aerosol medium is a simple, non-invasive way to take a look at public health and to do some occupational exposure biomonitoring. And then a little bit later, based on some of this other work, the pre-COVID work, we have started taking a serious look at the scientific rationale for using simple masks, which is kind of the inverse of our original work, where we used the mask as a sample medium. Now we're taking a look at the masks as a way to keep the aerosol from spreading. So to address both these kinds of issues, we've written these two editorials that I have provided the reference to at the bottom of this page. Another big issue that we have is, of course, the holy grail of much of our breath research, and that is to develop non-invasive preclinical diagnostics. So what do we mean by preclinical? Well, we would like to be able to diagnose disease state before it becomes a clinical condition. When you catch things early, you have a better chance of fixing them or resolving them. So we have efforts underway to develop screening diagnostics using gas phase, EBC, and EBA. One of the things I want to point out is these breathing methods are much less invasive than the nasal swabs. If you know people that have had nasal swabbing done, they find it unpleasant. If we could redirect some of these analyses to just breathing out into a real-time instrument, for example, a mass spectrometer, and getting some very quick results, this would be much more palatable to subjects and patients. And even if we do use the masks or the EBC, again, this is still relatively non-invasive, and these kinds of samples are much easier to transport than if we were to collect whole air or whole gas samples. And so there is some value not only to real-time analysis, but also towards offline analysis, but using EBC and EBA rather than gas phase samples. So to kind of get a handle, we have to start taking a look at what's out there in the literature, what's being done. And we are now taking a look at some references or some articles that people have written. And two that come to mind quickly are some perspectives that have recently been published in the journal. The first is one about using COVID-19 PCR and exhaled breath condensate. And the second one is one where they are looking at semivolatiles as fast diagnostic markers. These are representative articles, and I've listed them here, but there are plenty more out there in the literature. These just happen to be some good examples from the Journal of Breath Research. We want to restrict viral spread. A big issue is the mask. There is consensus that masks restrict the spread of COVID. It's been shown over and over that places where they have strict mask protocols have less incidence rates, etc. The consensus overall is that the SARS-CoV-2 virus is spread via the exhaled aerosol, and that the secondary transmission from touch and then touching the face or from surface contact is most likely from settled aerosol, although it is possible to have some form of human biological liquid transfer to surfaces and then transfer from hand to mouth. Everything is possible, but for the most part we consider that most of the transmission has its origin from the exhaled aerosol. Various bits of research have shown that the facial coverings, the standard hospital masks, and all manner of respiratory surfaces collect aerosols. We have actually done some of this research in our very own laboratories before retirement, in my EPA laboratories, at the university laboratories, and a lot of other places. I've worked with NASA and I've worked with the US Navy, aviators, and in all cases, when we have sampled or swiped or swabbed the insides of respiratory gear, we have found human aerosol remnants. That said, even prior to COVID-19, we were already using hospital masks and other respiratory surfaces as a sampling medium. And as an example of this, consider a paper I've listed below, where we worked together with an outfit, Canada, that had trained dogs that would sniff hospital masks that were worn by different people, primarily firefighters. And the dogs were trained with case control samples to sort these masks into cancer and not cancer. Now, before anybody gets all excited here, we don't know if the dogs are right. This is a preclinical analysis. If we already knew who had cancer, then we wouldn't need the dogs or the instruments. But the dogs were trained with case control samples, and then they were exposed to these unknown samples that may or may not have had preclinical cancer signs. The dogs could very consistently sort these samples into bin A or bin B, yes or no. We had the opportunity then to get two subsets of these masks, one triaged as yes by 4 dogs twice, and one set triaged by 4 dogs as no's twice independently. So pretty sure that the dogs were reproducibly sorting these. And then we took them, we ran them through a gauntlet of analytical methodologies in the lab, including semi-vol non-targeted analysis of using LC Orbitrap, using GC-mass spec, and using immunochemistry methods. So this turned out into a pretty interesting paper, and I've listed it at the bottom of this slide for you if you're interested. We have also been very concerned with analytical technology, especially with respect to identifying new chemicals in breath. So With this in mind, we've been taking a look at non-targeted methodologies to try to get a handle on compounds we may not be aware of yet that might be important for triaging preclinical disease from normal controls. When we compare these case control samples, we often find a difference in patterns, and we attribute these differences to being kind of related to the disease state. So you have a group of people with disease A and a group of people that are controls, nominally healthy, with no diagnosed disease. You compare kind of in a big group cases versus controls, what kind of compounds show up in the case, what kind of compounds show up in the controls, overlay them and start to sort out some differences. These differences presumably would be indicative of the disease state. So we can do this. And now we have to determine, is this a valuable thing to do? The question is, of course, that the differences could be from some reason that isn't the disease, just poor choice of controls or whatever, or differences in the groups that you don't know about. So in order to kind of circumvent this issue, it's good to know exactly what the compounds are so that we can say, well, these compounds make some sense because they fit in with some notion we have of the biochemistry of the disease. If the compounds are not somehow linkable to the disease, you may take a second look and go, well, maybe something else was going on here. So how do we do this identification of compounds? Even in the most complex instrumentation, you will have multiple candidates for every little feature that you find. We have now started taking a look at the chemistry of features by running various parameters through these big, what we call dashboards or big databases. So some examples are the EPA CompTox dashboard, PubMed, of course, where you do abstract searches for chemicals and such, and ChemSpider, which is a chemical configuration database that has a lot of information on how different chemicals are used, where they occur, what their metabolites are. These are all valuable tools once you get to the point where you need to identify compounds that you only have a vague notion about what they might be or what kind of class they may represent. So we started taking a look at this a while back, and at the last IABR meeting in Loughborough, England, we rolled out a presentation regarding the CompTox database and how to use it. We also wrote a short perspective on the use of a centralized resource for identifying chemicals. And we published this in JBR last year. And for your entertainment, I have listed it down here below. We are currently also working on a tutorial as to how to use the CompTox dashboard for a whole variety of purposes that are important in identifying compounds that are as yet unknown. Along those lines of using the journal to kind of collate information regarding COVID, we are now developing 2 new special issues for the Journal of Breath Research. Special issues are basically collections of articles with a particular theme. The first collection, which is being edited by Makoto Sawano from Japan and Jonathan Beecham from Germany, both are on the scientific board of the journal, and Jonathan is also on the editorial staff. We are encouraging people to submit articles that fit into this theme, and then we will collect them into a special issue to provide kind of a collated, valuable grouping of articles that would be a good starting point for doing some research as to what is the state of the art in breath and COVID. Similarly, we are developing a special issue for the physics of breathing, which I mentioned before, flow, pressure, volume, and time, where these serve as the dependent variables as a complement or replacement even for tracking health state instead of chemistry, chemical compounds, etc. In the past, we have downplayed this, thinking that it's more of a physiology basis, but I really do believe that the physics of breathing, the flow, like I said, flow, pressure, volume, and time, are important for our journal as well. And I'm hoping that by encouraging folks to submit in this field, we will develop the physics of breathing as a separate entity or a complementary entity for the journal and include it then in the scope. In case you want to submit here is a little plug for these submissions. You can contact us at JBR at IOP Publishing here, or you could just go to the website, which is also listed, to get more information. So in summary, the Journal of Breath Research is really trying hard to stay in the forefront of important COVID-19 and related diagnostics research. We're going out of our way to recruit articles, make special issues, to do as much as we can to become a central resource for this topic area. In so doing, we're also encouraging condensate and aerosol research in breath. I think this has shown to be a valuable resource in addition to gas phase analytics. And we are, of course, as I mentioned before, expanding the focus of breath research to the physics of breath, the flow, volume, pressure, and timing as dependent variables, and also starting to address ventilator use and monitoring of health state by using the physics of breathing as a complement to chemical analyses. And finally, cannot mention this strongly enough, we need to be careful out there. We have now recognized the needs for increase in documentation of safety precautions for all breath-related research, and the journal is doing its best to make sure that people who do submit to us are aware of this and hopefully have taken proper precautions in their own laboratories.
Fantastic. Thank you so much for that. I think there was a lot of really fantastic points, and what you finished on there, I totally agree with. It is more important than ever, for the field to be careful about the protocols used for collection and analysis of breath samples. And we've done quite a lot of work with our receiver device there, and the types of validation testing and the protocols that we've put in place to help steer some of the users there as well. So I totally agree and concur with that. So one of the questions that's been posed: what do you feel will be the largest long-term impact of COVID-19 for the breath research field?
I mean, impacting the research itself?
Yeah.
I really think that the major impact is the realization that breath has to be treated with universal precaution or pandemic precaution, just like blood during the AIDS and urine, which has always been treated that way as well. I've been doing breath research for about 30 some odd years, and we never once thought that we had to be super careful with breath. I mean, we have people sitting in the lab or in the clinic or whatever, breathing, and everybody else is breathing. And, sure, we throw away mouthpieces because we don't want, live contamination and things like that. We're very careful about the fluid transmissions, but nobody ever gave a thought to breath as a vector, unless you were dealing with, say, TB patients or whatever, right? And so this is the big change. This realization that we can no longer treat breath as like this throwaway fluid. And that's tough. I mean, I said in there, the average human breathes out about 16 cubic meters. And I don't really know how big your office is, but it's kind of a big closet, small office kind of. That's a lot of volume. And we completely ignore it. The breath is completely invisible unless it's a really cold day out. And we would never treat blood like that, right? Or, you can just imagine just throwing samples of blood around in your lab on the floor or whatever. You would never do that. And so this is the big game changer. We have been hit in the head about the fact that breath is now something that has to be treated much more conservatively and much more carefully.
That's really interesting. And I think one of the things that we've seen is that, everyone now knows that breath is important. And actually, a byproduct of that we've seen is people now see it as something that they want to actively research and study. a lot more. So I think COVID has we've certainly seen that
Oh,
kind of change in priority as a consequence.
yeah, I can really see that. And that's why at the journal we're going, we should become the resource for this. Rather than spreading articles into a variety of physiology journals,
Yep.
toxicology journals, environmental health journals, virology. I really like the idea of trying at least to build a centroid of research so that people who are interested can start there and say, oh, okay, here's something I might be able to do, or right.
Yeah, no, absolutely. I think that broadening really makes a lot of sense. For the non-volatile fractions, so there's EBC, EBA type capture approaches. Obviously, feeds with EBC, there's a bit more complexity there in having to condense. Do you feel if there were going to be a solution for a capture that may be fieldable in a relatively near-term cost-effective way, is that more likely to be an EBA type approach or an EBC approach in your opinion?
Well, personally, having done both for many years, mostly EBC, which is in the Venn diagram, EBA lives inside the circle EBC. But the reason I started looking, or I shouldn't say we started taking a look at EBA was a complete accident. In the clinic, in the labs, at the university, we would just always go with EBC. We had machines. Terrence and I developed some instrumentation to do this with timing and volume and all that kind of stuff. And that's what we did, because that was very precise and well controlled, and we understood it really, really well. What happened was I started working with firefighters and pilots and people like that, where you can't do that. These guys are, putting out a fire and using axes or flying an airplane at 30,000 feet. So what do you got? Well, they all wear respiratory kind of gear. And I actually spent one summer going to Navy bases for F-18s, which are high-performance aircraft in the US, carrier-based aircraft. And I would swipe their masks after they were done, because we wanted to see if some of the fluids from the aircraft were actually reaching them through the breathing system. And so we're doing this, we're going, oh, okay, we can do this. And we're getting the aerosol from the airplane in there. And we're also getting human aerosol, right? And so we did a whole study on the emission from the body during flight, during firefighting, during, used a variety of respirators and ventilators, respirators, sorry. And we found, we focused in on human cytokines, which pro-anti-inflammatory cytokines, which are to some extent biomarkers. Well, they're biomarkers. What they are biomarkers of is still out for debate. There's high variability between people, also within people. But the point was, we could see human aerosol unambiguously. Immunochemistry will not show you a dog cytokine. It will not show you a chicken cytokine. If it's a cytokine that's got a receptor for human, it'll be human, right? And so that was an epiphany that these surfaces might be valuable for at least getting a qualitative look, not only at things that you've been exposed to, have gone in and come back out wet, but also things that you made on your own, right? Single molecules. So that's how it started. Then, I talked a little bit about that dog experiment, and I did appreciate the other dog talk earlier today, where the people out in Canada, called cancer dogs, cleverly enough, have been working with firefighters across the US for dozen years. And they're, these guys just put on these medical masks, They fill out some paperwork, which is designed to take about 10 to 15 minutes, and then they take their little mask and shove it in a Mylar envelope and mail it up to the dogs. And there are good anecdotal results from that. Now, is it pure science? Maybe not, but it does provide a service in that if the dog says, hey, you might want to go to the doctor, and the guy goes to the doctor and the doctor says, oh, on this scan, you show some preclinical cancer. Those kind of stories are powerful, right? And I wanted to see, what kind of compounds are the dogs possibly sniffing? They can't be volatiles. I mean, this mask has been worn, it's been stuffed in an envelope, it's been shipped to Canada, but they are obviously smelling something. And, we basically got the idea that it might be the semivols and the way dogs breathe. I don't know if, I don't think that was discussed earlier, but they create a turbulent zone. If you take a look at the face of a dog, he's got like the nostrils and the little slits. Well, he creates a cloud of turbulence in front of him that rattles things loose, basically. We don't breathe like that. Sensors don't breathe like that. But the dog, he snorts out through these slits, and he makes a mess in front of him, about 50 centimeter turbulence zone. And so we thought, well, there is maybe something to this aerosol issue, because the dogs seem to be able to sort. We then threw, all the high-end instrumentation at it, and we found better discrimination at the heavier molecules using LC Orbitrap than we did in GC mass spec and immunochemistry as well. And so that was, it's another good path. I love VOCs, don't get me wrong. I've been using VOCs. I started with the agency as a contractor in 1979, measuring VOCs in the air, in migrated air, breath, etc. But there may be something to this. Going into the heavier compounds, which we know do come out, right? So anyway, that's my soapbox there.
Yeah, no, that's very interesting. Actually, in explosive detection, so obviously, very low volatility there are some of its particulates. So one of the approaches was to essentially try and liberate the particulates with little puffs of air as well that can then go into the system that, apply heat, do a bit of desorption in it to get it into that volatile space. It's interesting as well, like when we first started considering the exhaled breath aerosol, we were lucky in one regard because we had put a filter material in the mask of our receiver device, essentially to stop any type of cross-contamination into the base unit. There are still the same types of materials you find in N95 masks. So by virtue of the fact that that was such a good capture device to stop contamination, it then acts as obviously a capture device that you can do subsequent analysis on. So as Mark touched on earlier, we've obviously done some of the lab-based aerosol work there, but hope that we'll be submitting to the journal in the very near future with some of the clinical samples when they come through. We've got time for one last question, which I think is really interesting. So obviously with the swab itself, when you look at the combined swab PCR, there is a bit of an issue of false negatives there. PCR itself is obviously very reliable. So the question that's being posed here is, is the problem potentially the nasal swab itself? So if you're not hitting the right point, you're not getting the virus, you're obviously not going to get the positive hit there. So might, breath condensates or exhaled aerosols be a more homogeneous sample and actually give you better performance from a false negative standpoint compared to the swabs?
Yeah, so I agree with you. If you get punched in the nose when you're a kid, that swabbing you may not even be reaching a place that had breath in it, right? Deviated septum, things like that. So there are those issues that may or may not be addressed. When you breathe out through your mouth, through your nose, whatever, you always make aerosol. You can't help it. It's always there. And it has multiple sources in the body, and there's papers written. I, I part of it is the turbulence in the upper airway, and the fluid linings kind of get disrupted. But then there's also the mechanism, what's called surface film disruption in the alveoli. The alveoli are little balls, and when you breathe in, they pop open, and there's a little surface across the top, and it makes a little bit of aerosol. And when you breathe out, you sweep it out. Again, some people say A, some people say B, some people say both. But the point is, it happens well below the nose and mouth. And so the aerosol fraction then is representative of these mucus and watery linings in the lung, which, if you're sick, would conceivably contain the virus and other things. And the aerosol itself gives it wings, right? So yeah, I agree with you 100% that if you have a good method, it is more likely to be universally valuable than the swabbing, right? I think. Now, this is not as well developed as the swabbing, and so, or the blood test. I think they're using some blood tests as well. But yeah, in the long run, I think that is the path to go. Now, one thing I want to caution is using surfaces to crash aerosols into is qualitative. We really don't know how quantitative it is. And I would think that would matter, on mask fit and things like that, as well as, the layering. But the one thing I do want to say is, unlike the N95 masks, which basically give you bruising around the face after a while, and it forces the air in it, well, depending on if they have the vent or not, but it forces it through the material. Yep. The kind of masks that people you see in the grocery store wearing, at least in the US, are not like that. They're literally, you can feel the air leaking out all over the place. It fogs up my glasses, for example. But there is the physics of the aerosol that causes the aerosols to go straight when the gas goes elsewhere. And this is the mechanism that's important, because it crashes things out. However, anyone can, of course, say, well, how effective is that? Good question. Varies, I'm sure. But if nothing else, it does help because of the physics of aerosols.
That's interesting. So obviously you're relying on the impaction. So it's the heavier droplets or aerosols, but obviously the smaller aerosols, have a chance to actually get out and make it out from the deeper airways.
Consider how big the viral particles are, and they have to be encapsulated. I mean, they're pretty big compared to an oxygen molecule or a CO2 molecule or a nitrogen molecule. So you've got like factors of 1,000 things going on here. I would propose, I mean, I've tried to look this up. Again, there's a little bit of controversy here and there. But people have done aerosol sizing. So it's not going to ever be a perfect solution. Again, I always looked at it from the perspective of a sampling medium, rather than providing a safety measure. But I do see how it does provide a safety measure, and the anecdotal or the observational evidence is there. Places where they use masks have less infection rates. You can't get around that. You go to a movie theater where nobody's wearing masks, and you may have 20 cases of COVID out of there, right? Or other kinds of gatherings. And so I propose that the mask wearing works. And the reason that it works is for this kinetic impact of wet things. Once they dry, you may not have that anymore. So this is why the masks are an altruistic kind of activity, in that you are essentially keeping your aerosols from leaving you, drying, and then getting somebody else. So I do believe personally that, or well, I did study physics a little bit here and there. And at EPA, we also use various virtual and impactor kind of surfaces for sampling air. So we have a pretty good track record on understanding the laminar flow and the turbulent flow and the kinetics of particles and things like that. So, all your PM 2.5 samplers have these impactors with a little bit of grease on them, and the heavy particles smash into them. It's the same concept.
Yep.
It's nothing magic here. Excuse me.
Excellent. Well, thank you so much. That was fascinating talk and discussion. Again, there's a lot of really exciting research happening, not just in COVID, obviously in the broader breath field as well. We all know where we've got to go and publish the papers with the expanded scope.
Yeah, listen, and I want to thank you for putting this on.